Mr. Winchell here. I hope you guys are all doing good. Wish school year hadn't been canceled, but I'm going to try to upload these videos every time we do an assignment to give you guys an example of kind of what to do and how to do it. So today I kind of want to go over some Punnett Square basics and then show you guys how to do a dihybrid Punnett Square. Okay, so come over here. Maybe you can't read my handwriting, but it's all on the PowerPoint. So first I just want to explain a couple vocab words. So genotype. Genotype are the letters that we use to represent the alleles, which are the different versions of genes that a person carries. Now, all the examples we do are going to have two letters. They could be two capital letters, one capital, one lowercase, or two lowercase letters, and that's because we are diploid. We get one gene, one copy of each gene from mom, one copy from dad. Okay, so the genotype are the letters, which represents the genes that we carry. The phenotype is how those genes are physically expressed. So like this plant and this plant would be represented as tall, and this plant would be represented as short. So as long as you carry one of the capital letters or the dominant allele, you express the dominant trait, which for pea plants is tall. And if you have two recessive alleles, which are lowercase, you express the recessive trait, which in this case is short. Okay, now when you set up a Punnett square, you set it up like so. The typical Punnett squares are just 4 blocks. You put the parents' genotypes, the parents' gametes on the top and the left just by convention. Now, you split the letters one on each block. The reason being is these represent the gametes, the sperm or the egg. Now remember, during meiosis, the gametes or the genes get separated from each other. So this represents one egg could carry the big T or the little t. Well, over here, let's just say this is the male. Each of his sperm carry the capital dominant allele. Okay, so when you fill in the Punnett squares, you take the letters on top and you just drop them straight down into the blocks. The letters on the left-hand side, you carry them straight to the right. Now, I want you to notice I put the capital letters always in front of a lowercase letter if they're found in the same block. Now, what do these blocks represent? Each one of these blocks represents a quarter or 25% chance of the offspring having that genotype. So basically, this is simulating sex. Okay, so if these two parents had a kid, they had a 1/4 chance it would come out like this, 1/4 like this, 1/4 like this, 1/4 like this. Which one would happen? We don't know, but we can predict up to 25% chance probabilities. Okay, last thing I'm going to show you before we do the practice problem is how to do FOIL. So we're going to see this genotype in a second on the assignment I go over. I already wrote out the parent's genotype. I want you to notice there are 4 letters now. This is called a dihybrid Punnett square because we're going to be looking at 2 genes. We're going to be looking at the gene for plant height, big T representing tall, little t representing short, and then A's are going to represent where the flowers are found, whether they're found on the ends of the flower or the stem or like the middle. Okay, so that's why there's 4 letters. It's 2 different genes here. So to FOIL, you do, it's called first, outer, inner, last. First means you take the first letter of each type. So big T, big A. O represents outer, which means the 2 on the outside. So big T, little a. I'm going to put a comma in between them. Okay, then I represents inner. So it's going to be the 2 on the inside, which is little t, big A. And then finally, L represents last, which means the second of both types. So little t, little a. These guys represent here the possible gametes that this person or plant could make. So this plant's sperm or egg would have a 1/4 chance carrying this, 1/4 this, 1/4 this, 1/4 this, and so on. Okay, so now I'm going to walk you through the first problem. So it says in garden peas, tallness, capital T is dominant, and shortness, lowercase t, is recessive. Axillary flowers, or capital A, are dominant to terminal flowers, which are going to be little a. Okay, so what are the expected genotypes and phenotypes of the offspring? So we're going to use the steps, which I showed you in the PowerPoint. First, we're going to make a key. I'm not going to make a key on this one because the key is kind of already given to you in the problem. But if the key is not given to you and it doesn't tell you what letters to use, I would always start with that. Okay, step 2 is find the parent's genotypes. So it says parent 1 is heterozygous tall, heterozygous axillary. Now, hetero means one of each letter. So heterozygous tall means big T, little t. And then heterozygous axillary means, again, the same thing, one of each letter, capital A, lowercase a. So that's the first parent. Okay, then we use an X, not as a multiplication sign. It means crossing. It means taking It means the plants basically reproduce with each other. Okay, the second plant is heterozygous tall, terminal plant. So heterozygous tall, again, means big T, little t. And terminal, it doesn't tell us homozygous or heterozygous. When it says terminal, if you look up here, it says terminal is used with lowercase a. That's the recessive trait. The only way the plant could have terminal flowers is if it carried both recessive alleles. Okay, so that was step 2. Now, step 3, we're going to foil just like we did right here. Now, if you notice, the example I gave right here is the same as on this problem. So I'm not going to walk you through that one again. I'm just going to write out the possible combinations, which again, that means this plant could have these types of gametes. Okay, now we're going to do the same thing for this one. So first, big T little a, outer, big T little a, inner, little t little a, and then last, running out of room here, little t little a. Now, I want you to notice that there's 2 big T big A's and 2 little t little a's. If you have 2 that are the same as each other for the same plant or same parent, you can cancel them out. Now, I can't cancel ones out over here with ones over here. These are separate plants. These are this plant's gametes. These are this plant's gametes. Okay, so now I see I have 2 here and 4 here. Now I'm going to make my Punnett square, which is step 4. Now, because I have 2 and 4, you're going to make a Punnett square that is 2 by 4, which means 2 columns or 2 rows, and then 4 columns or 4 rows. Either way, I could have turned it like this and it would have been the same thing, but I always like to do mine horizontally. Okay, now these 2 are going to go on the side with 2. So big T, little a, little t, little a. Now these are the 4, so I'm going to put them up top. Big T, big A, big T, little a, little t, big A, little t, little a. Okay, now I fill in my Punnett square. I recommend doing these ones one at a time. So I look over here, we got to keep the letters together. So I have big T, then I have big A, little a. Again, make sure you try to keep the capital letters before the lowercase letters, but keep the T's together and the A's together. So I continue, big T, little a, little a. Big T, little t, big A, little a. Big T, little a, little a. Big T, little t, big A, little a. Big T, little t, little a, little a. Little t, big A, little a. Finally, little t, little a, little a. Okay, now last step is step 5, which is where I analyze my results. So I want to look through here, and at least I'd like you guys to find the phenotypic ratio, which is kind of what these plant offspring would look like. Okay, so I'm going to go through. This plant has a big T and a big A, which means it's going to be tall, and it's going to have axillary flowers. I'm going to go through and count how many have a big T and a big A. 1. Here, I'll put a star on them. 1, 2, 3, and that's it. So over here, 3 out of 8 are going to be tall, axillary. Okay, now I'm going to go through and see this one is tall because it's a big T, but it has 2 little a's, which means it is terminal. Okay, so for these ones, I don't know, I'll use a triangle. Okay, so big T, but both little a. So 1, big T, little a, 2, big T, little a, 3, and neither one of those. So again, 3 out of 8 in this case will be tall, but they will be terminal flowers. Okay, so I have 2 left. If I look at this one's 2 little t's, which means it's going to be short, but it has a big A, which tells me it's going to be axillary. Okay, I'll use a circle as my symbol for that one. So this one is going to be 1 out of 8 is going to be short, but axillary flowers. Okay, and then lastly, I need some other symbol here. I'll use a square. This one has 2 little t's and 2 little a's, so it's going to be both recessive traits, which in this case is short and terminal. So again, 1 out of 8 is short, terminal. Here, let me put the symbols next to them so you guys can see where they came from. Okay, and that's it. So those are the dihybrid Punnett squares. I know they're a little bit of work. They're not always 8 blocks like this. They can be up to 16 blocks, which takes a while, but sometimes these ones all cancel out and you're only left with 1 block or 2 blocks or even 4 blocks. So this was one of the longer types on the assignment. Please give it a try, and then if you have any questions, let me know. Thanks.